Hello, everyone. So what if I told you that I can make things disappear? Just like that. No, not really. I just learned that for today. Hello, everyone. My name is Yumi, and for a few months of my life, I was dedicated to turning bodily tissues transparent. So what do I mean by that? It's kind of like turning this mouse, right here, into something like this, where you can see through the entire grid. We call this tissue clearing. You're probably wondering, but why? Why on earth we would do this? Well, by making tissues transparent and then highlighting all the things that you want to find, you can make very, very accurate 3D images of entire blocks of tissues, organs, or as you've just seen, entire bodies. So our bodies and tissues are obviously inherently 3D, so it's really important to understand their 3D structure. But currently, we don't really know very much about the detailed 3D structure of our bodies just yet. We know a lot about our bodies on a larger scale, like entire organs, or maybe tendons, larger blood vessels, enough to make a medical student like me cry. But to improve our understanding of biology and disease, how diseases progress, and therefore how maybe we can potentially treat them in the future, we need to understand diseases and our bodies on a much smaller scale, at the level of individual cells. So imagine I give you a cube of tissue just like this, and I asked you how on earth you would find out what's inside. Well, you could cut it open, right? And that's the main way we did this before. We would cut it into many, many thin slices, like an onion. And then we would image each one and reconstruct the cube again by putting all of those images together. As you can probably imagine, this is a very, very tedious process. You might have to prepare hundreds, if not thousands of slices. And before you can even image them, they might be torn, they might be folded, stretched, or otherwise damaged, causing a lot of information loss. So you're not going to get the most accurate data. Wouldn't it be much easier if the tissue was transparent like this, except for what you were looking for? You could just kind of find the stuff that you've highlighted already, like blood vessels, neurons, cancer cells, or anything else you're interested in, and then kind of ignore the rest of the tissue. So it's kind of like reading an annotated textbook. Instead of reading the entire thing, hundreds and hundreds of pages, you can just find the important stuff and ignore the rest. All right, so to understand how we make tissues transparent, first we have to understand why they're not transparent in the first place. You've probably had a lot of thoughts about your body before, but you probably haven't looked at your arm or any other part of your body and thought, why can't I just see through this? Well, there's three main reasons why. The first one is pigments. So some tissues have way more of this than others. I'm sure you can think of examples like red meat or even liver. And I'm sure you also know of a very, very easy way to get rid of pigments: bleach. But not so fast. Remember how you need to highlight the things that you want to find? If you just bleach all of the chemicals, then you're not going to see anything at all. You need something that will preserve your highlighting, but also get rid of everything else that's distracting it. So it's a bit closer to color-safe bleach. So the second reason why tissues aren't already transparent is something called autofluorescence. Now, this is a science term to explain how certain substances, if they've absorbed light already, will glow. They will give off their own light. So it's kind of like these glow-in-the-dark stars you might have had in your childhood bedroom. One very common example is collagen, the stuff that makes your skin more elastic and smooth. So yes, your skin really does glow. No makeup or vampire blood necessary. This really isn't a problem if what you are looking for already happens to have autofluorescence, because you can then exploit that and just kind of find it. So for example, in this case, they used this green glow of mouse brains to highlight all of the neurons that they were looking for already. But if they don't have that, then that's just going to distract from what you're looking for in the first place, all of that beautiful highlighting you've already put in your tissue. The third reason why is something called refractive index heterogeneity. Now, your bodies, even your cells, are made up of many, many different things. These can be roughly sorted into three categories: protein, fat, and water. Now, these things aren't distributed randomly and evenly throughout your tissue. They're organized in a certain way, like obstacles in a pinball field. And just like the obstacles can change the trajectory of the ball, these substances will bend, that is, refract light, to different degrees as well. So in technical terms, The degree to which something bends light is called the refractive index. So for a tissue or anything to be transparent, the light needs to be able to pass through it in a straight line, just like this. But with all of this water, lipid, and protein in the different parts of the tissues, it's not going to go straight. It's going to bounce around a bit like this. So the main part of tissue clearing becomes equalizing the refractive index throughout the entire tissue. That way, the light that's getting bent like this gets perfectly reflected by other light that's also getting bent. So the only light that's left is that which travels straight. There's many, many ways to do this. So remember how I said that there's three main components, water, lipid, and protein? One way to do this is to replace, for example, the water here that's shown in blue with something that has a higher refractive index than water. But you can also get rid of both the water and the fat and kind of replace those with other things. Or you can actually decrease the refractive index of the tissue by soaking the tissue in many water-based substances. And then getting rid of the lipid, which has a higher refractive index at around 1.4 or something, and then kind of messing with the proteins a bit to decrease their refractive index as well. So based on the refractive indices of many different chemicals, as well as their various tissue clearing properties, like the decoloring I mentioned earlier, we shortlisted some chemicals. And then we experimented to find out what was the best concentration of each one to use and which combination of chemicals would work best. We did this through side-by-side comparisons of the same slice of brain to make sure that they were the same size. And then we tested which one would be more transparent and which one would preserve fluorescent dyes the best. Simple, right? Well, no. There's many other things that scientists should consider when they're coming up with a new tissue clearing method. So for example, are the chemicals expensive? Are they toxic, requiring lots of different safety features? Are they easy to prepare? How quickly should they clear the tissue? Should they be really, really good at clearing a certain kind of tissue like bone, which is very different from, for example, your skin? Or should they be able to clear the entire body relatively well, like a jack of all trades? What kind of staining methods should the tissues be compatible with? And so on and so forth. So all things considered, there's actually quite a lot of variety in the tissue clearing methods that have already been published. And this is the result of many, many teams' hard work and dedication. So now scientists can basically just pick which one will work best for their uses and go with that. And while tissue clearing is a relatively young field, we've already come up with some very, very interesting findings. So for example, Morgan Bell's team was able to trace all the peripheral nerves in infants. This has allowed people to understand more about how normal infants develop, and therefore we can take this information and look at, for example, how infants with disease develop, and therefore understand a bit more about how the disease starts and progresses, and therefore we can start to think about, for example, how we can potentially treat these diseases. The same team has also come up with a very extensive view of all of the blood vessels in an 8-week-old human embryo's hand. Tissue clearing has also been quite important for cancer research. So for example, Pan Chen Chen's team was able to look at where the cancer has spread to in a mouse, and also whether or not the treatment has reached those cancer cells. So here we see all of the cancer cells that have been reached by the treatment, which was a specific antibody in yellow, and all of the cancer cells that weren't reached by the treatment in red. In this way, we're able to understand a bit more about how treatments may or may not be effective and start to understand how to improve treatments a bit more, or which treatments are better than others. Tissue clearing has also been really important for bone marrow research. So here, Maximilian Gorilashvili's team has cleared mouse femurs. And we can see here that the blood vessels are shown in red. The megakaryocytes, which are cells that produce platelets, are shown in green. And the white is just the bone. So through this research, we found that previous 2D imaging has shown that the size of megakaryocytes was relatively small, but now we understand that they're actually a lot bigger than the 2D images led us to believe. We also understand that the distance between megakaryocytes and the blood vessels that they secrete the platelets into is actually a lot longer than we thought as well. Tissue clearing has also been really important for neurodegenerative disease research. So in this case, Anthony Mataral's team was investigating Alzheimer's disease in mice. And they wanted to see if we could treat Alzheimer's disease by giving the mice a combination of auditory and visual stimulation. And so they had mice that had the stimulation and mice that don't, both that were affected by Alzheimer's disease. And then they cleared the brains of both groups of mice to see how much amyloid beta was in both groups. And we saw that these mice with no stimulation actually had more amyloid beta, the protein that accumulates in Alzheimer's disease, which is the white specks that we see here. And then simultaneously, they also found that the mice that got the stimulation actually not only had lower levels of amyloid beta in their brains, they actually showed improvements in their cognitive tests. But there's still more work to do right now. We still don't know about the details of how tissue clearing works. So for example, I can't tell you right now that chemical A is much better than chemical B, even if they're very, very similar, but they have slight differences, and I can't really translate those differences into telling you which one would be better for you. So right now, the tissue clearing experience is much more of a trial and error process to figure out just what works and what doesn't. But maybe, if we keep trying, we'll eventually find out. And hopefully, tissue clearing will make us see things that we haven't seen before. Thank you.